Welcome to RegenMedGlobal, the podcast, where we bring you the latest breakthroughs in medicine and research to help bring hope in fighting incurable diseases. In this podcast, we're discussing the recent developments of novel approaches in using AI to improve the way we identify cancer, particularly in the bile duct and thyroid. Behind these developments, we have PhD candidate at Case Western University, Shayan Monabbati. Shayan, it's a pleasure to have you. Before we dive into these topics, tell us a little bit about your background.
I'm a final year PhD candidate in systems and control engineering at Case Western Reserve University, and my focus is on the biomedical space, and particularly in the field of digital pathology. And it's a relatively new field that kind of gained a lot of traction back in 2005 with the emergence of AI in alleviating the workload for pathologists, right? So what is digital pathology? If you think about it, it's just the compoundment of two words, right? Digital and pathology. It's exactly what you think it is. It's digitizing the realm of pathology, and where traditionally pathology is done with standalone microscopes. And so what pathology is actually probably get this nailed down right away at the beginning is looking at or the study of disease, or examination of tissues or cells under a microscope. So my research is at the intersection of computer science, statistics, pathology, and machine learning. And so what I'm doing now in my thesis and work is to use artificial intelligence to quantitatively mine patterns on routinely acquired data. And so this data consists of digitized images of histopathological and cytopathological tissue samples. And so we use this data for a variety of different clinical outcomes, right? So we use it to diagnose, to prognose, and predict treatment for patients. So there are only about 21,000 pathologists, according to the AMA, in the U.S. So think about, the ratio of how many patients there are to a doctor, right? So how many of these patients have to go in routinely for biopsies, or for example, let's say if you're a woman who's 50 years old and you have to start going in for mammograms every 5 years, the oncologist would have to send that sample to the lab, and then a pathologist would look at that sample and then give a diagnosis to see that do you have healthy tissue or do you not have healthy tissue, right? So again, I'm talking about the continuous feedback loop of how AI and doctors work in tandem, right? So a lot of people have this misconception that AI is going to replace digital pathology, or AI is going to replace doctors. Right, and this has just been it's been a farce for about 30 years, right? You can go back in the 1980s, where there were actually startups who tried to digitize and to create these imaging models on digital mammograms, right? And so 30 years later, we still have our traditional methods of people going into the doctor's office, and the pathologist is still there. To give that patient and clinical interaction, right? So I don't believe this thing that AI is going to replace them. If anything, it's about augmentation, how AI can help or guide really the areas that pathologists are currently struggling with.
Awesome, very cool. That's and when did you start doing all of this in terms of getting into digital pathology?
So I guess my journey with this particular field in digital pathology started. I mean, I said I'm in systems and control engineering, and you would think, well, what does that have to do with that? Well, I started my grad school in 2018 in systems and control engineering, and it's a very broad field, right? If you think about, it's just the study of systems and how they apply in engineering life, right? So a system is anything that requires an input and spits out an output, and everything in between is how you would describe your system. And so I saw actually my advisor's face on one of the campus newsletters. I think one of the first few months I was starting as a PhD student at Case, and I saw, wow, this is something I've really been interested in a while. Kind of getting into the medical space. I've always been interested in the medical space, but kind of had this journey throughout my undergraduate career. So I've done research from since I was 18 all the way up until now. I'm 26. Just 8 years of going through different medical fields and trying to identify where my niche is, where I belong. And I think I finally found it with this particular field. So I kind of just looked into the newsletter one day, and I saw my advisor's name, Anand Madabushi. And I looked into his work and said, wow, this is really, really interesting. And it is related to exactly what I was studying at the time, which was systems engineering. Because if you think about it, an image is a signal, right? And so a signal gets fed into a system, and it spits out another signal, an output signal. So I'm like, yeah, this is really, really interesting, and I can see how the nexus of combining systems engineering with the biological context is just really fascinating to me.
So Shayan, right now you're using AI for your various researches. Can you tell us a little bit about the different areas of cancer that you're using AI for in your research?
Certainly. So I work in 3 different projects right now throughout the course of my PhD. And they're all tackling different clinical outcomes, and they all look at different disease types as well. So the first one that I've been working on, and that's the first project that I worked with, and I also published a study on this in cancer medicine last year, is looking at an automated way of diagnosing malignant and atypical cell clusters on bile duct brushing samples. So it's a lot of word salad there, but essentially, how we collect data is to look at cytopathological samples. So we look at particularly cell clusters that are obtained from the lining of the bile duct, which is a kind of a network of small vessels that connect the liver to the gallbladder, and is responsible for carrying bile fluid. And so the cells within the lining of those bile ducts can develop cancer, and it's actually a very rare but aggressive form of cancer, very similar to pancreatic cancer. Of course, they're very proximally located, and so that was one of the projects, is to be able to create an algorithm that would automatically identify what are malignant cells and what are atypical cells that may look malignant but are actually benign in nature. So that's one, and the second one is looking at the papillary thyroid carcinomas. So this is the most common type of a thyroid cancer, and it's a very typically indolent disease. Not many people die from it, but what is an unmet clinical need in that space is kind of looking at the variability of the risk of recurrence within these patients, because there are so many different subtypes of papillary thyroid carcinoma that you have to treat them very individually and on a tailored basis, right? So there are some variants that are less aggressive, and there are some that are more aggressive, and our job is to be able to create an AI algorithm that would kind of characterize on the pathology slides, what the machine can see from the digital images are these disease patterns, right? So anything from looking at nuclear shape, how nuclei or cancer nuclei are kind of separated out from each other, how lymphocytes that are invading the tumor, how those are kind of scattered through the tumor microenvironment, and really congregating all of that different data that you can extract from these images together to create a model that is able to predict the time to recurrence for these patients. So just by looking at a patient's image, we're able to see, we're able to calculate what their risk of recurrence is based off of their profile. And what's actually interesting about this project is that we're expanding it by including genomic information as well. So we go back and we improve our model by incorporating certain genes that are linked to pathways that we've known from literature that are responsible for the proliferation of these papillary thyroid cells. And so we went back and we improved the robustness and the accuracy of our model doing that. That's a really interesting project with a lot of potential in that. And then lastly, we have the oral cavity project, and this is a one that's new, and I can't really disclose too much information because I don't want to get scooped on it. But we're looking at the combination of genomic information and again pathomic information. When I say pathomic, this is in reference to just quantitative data that we obtain from pathology, and so the combination of these two to identify hot mutation regions on patients that have tongue cancer, right? So it's really looking at the epigenetic variability amongst patients and throughout the patient or throughout the tumor itself within each patient.
I'd like to just start off with your recent study about the AI and the bile duct. Now, right now, for detecting cancer within the bile duct, you shared with us that it's fairly tough to go and have it detected. Can you tell us as to what makes it so difficult to be detected?
Yeah, so I would say that the number one issue with trying to detect these malignancies on bile duct cancer is a concept called inter-observer variability, and that is the pathologist's concordance or ability to kind of come to an agreement on a diagnosis for a patient. And the reason why some of these cases are so difficult to detect is, well, we have a myriad of reasons. One of them, it could be the staining protocol. It could be different from site to site. Sometimes in my own personal experience, I've seen images where nuclei and cytoplasm are very cluttered together. You have nuclear overlap, which is actually a cytologic criteria that pathologists use to identify malignant cells. But to try to delineate those nuclear boundaries from one another is quite difficult, right? So sometimes you could have like a collection of cells that could actually be atypical, but are actually malignant, and the other way around. So you have atypical reactive cells, you have atypical suspicious cells, and we have suspicious for malignancy, right? But they're actually benign in nature. So if you misdiagnose those particular cells and you come up with a wrong diagnosis for a patient, you could really ruin that patient's life, right? It's if you tend to overtreat a patient when they actually have a disease that may not actually be a cancer, or it could be a cancer but it's way less aggressive than initially thought of, right? You don't want to be able to affect that person's quality of life, right? Both from a financial perspective and from a healthcare perspective, right? We don't want to overtreat the patient with chemotherapy or radioactive therapy, and we also don't want to hurt their pocket, right? So I think around 90% of cancer patients lose their life savings within the first year of treatment. So we definitely want to be able to mitigate that issue.
Shayan, before we get into the details of the study, can you tell me a little bit about how many people, pretty much how many individuals you conducted within the study?
Yeah, it's a good question. So in any study that we had in an AI study, we always break down our data set into two sets, right? We have a training set and an independent validation set. So in our training set, we had 100 patients that we collected from the university hospitals here in Cleveland, and then also we had another set of 60 that we used for validation from from Cleveland. So we didn't really have the resources at the time to go out and get data from other institutes because it's, again, such a rare disease. It's really difficult to get data on this particular disease. And yeah, so we trained our model on 100 patients, which actually one image would correspond to one patient, and in the training and the validation studies sets.
You had those 100 patients. Can you tell us a little bit about what the ultimate goal was of the study?
Yeah, so the goal of this study was to identify within particular patients if they were malignant or if they were atypical, and that was done based off of cytology, right? So initially, a patient was looked at by a cytopathologist, and their biopsy was obtained, and then the cytopathologist went and looked at the cells that were in that patient's biopsy sample, and so they made a diagnosis with the help of other pathologists as well to make some kind of diagnosis if this patient is either benign, if they're malignant, or if they're atypical, which they're indecisive about, right? They need further maybe another method for kind of really getting the diagnosis concrete. So our job was to come in and create an algorithm that would be able to identify those atypical cells, and then of course as well be able to identify our classic benign versus malignant cells as well. So the difficult part of the algorithm was to identify those atypical cells that could either be reactive or suspicious. And so reactive is benign and suspicious is also benign, but the morphology of a suspicious cell cluster is that it looks like a malignant cluster, but it's actually benign. So to be able to identify those cells on our AI with a high degree of accuracy is ultimately what our goal of our study was.
Okay, now with the goal in place, how did you measure success within your study?
And so the metric that we looked at is AUC, which is area under the curve, and that is a metric that shows a trade-off between the sensitivity and specificity of our predictions of our model against the gold standard. And so the gold standard in this case would be new generation sequencing of these cells. And so that really provides the ultimate diagnostic standard that pathologists use nowadays to identify whether cells are benign or malignant or atypical. And so we compared our model's performance against the NGS standard, and in terms of its specificity, it was up there. It was almost at 98-99%. So it was detecting false positives quite well. But really the main issue, the clinical issue with bile duct cancer is looking at those false negatives, right? So things that look benign but are actually malignant. Right, so our job was to be able to increase that sensitivity from what it is now was 44%, and we got it all the way up to 72%, and that is with including atypical patients as well.
Is there any additional findings or anything else that stood out to you using AI for the sensitivity of cancer within the bile duct?
Yeah, so I could talk about really what our findings were, like the kind of the meat and the bones of what our AI actually found. And so what we were able to find is that features pertaining to the nuclear shape and texture of the entire cell cluster was very important in determining the difference between a benign and a malignant cell cluster. So things like the shape of the nucleus, the prominence of nucleoli within the malignant nuclei were very, very important in determining that difference between a benign and a malignant cluster. And also looking at kind of image texture is a very abstract thing, and to make it easier for viewers to understand is kind of looking at the differences or the changes in image contrast of pixels that are closely to related. So it's kind of looking, if you wanted to look at something like a black screen, there's no texture in that image because pixels around it are similar in terms of their color value. But if you have something with a high amount of texture and a high amount of a kind of energy and disorder, is what we use in the imaging world to describe that. These patients with these malignant characteristics had a lot of this disorder around the cytoplasm, and that was due to the actual collagen that was, or sorry, not the collagen, the chromatin distribution throughout the cell, right? So these patients that were malignant had a higher disorder of chromatin, like the chromatin were more concentrated around the nuclear boundary, for example. So it was really interesting to see that we seen things that we could see with our own eyes. That was kind of validating our findings of our AI models. So it's not just a black box approach where we just throw in our images and leave the burden to the computer to see what features are picking up the driving signal for this malignancy.
So were these incidental findings, or were these findings that you were looking to, were you looking to try to find these findings?
Very, very good question. So And this also goes back to the whole debate of deep learning versus traditional machine learning. So in this particular model, we stuck with a traditional machine learning method in which we already pre-select the features that we want to feed into their model. So we are actively looking for features on the pathology that we have already learned from 100 years of literature, that we've amassed all these criteria that show or have proven to show malignancy. So the question for our methodology was, what kind of computerized features kind of are associated with those biological underpinned criteria that we're looking for. So it's definitely something that we took into consideration during the process of building our pipeline, was to look at those features that were interpretable and had a biological underpinning.
Okay, so those features in terms of the characteristics of the cancer, those were already historically in literature known to be associated. Is that right?
Right. Yeah.
And so you and your team basically utilized that and applied it to the machine learning technology to help identify those and try to improve the sensitivity. Is that correct? Am I interpreting it the
Correct.
right way?
Yeah,
Okay.
and the advantage with using a machine over a human is that, well, if you think about what computers are really good at, they're really good at doing well-defined tasks over and over again, right? Almost flawlessly, and with on very large data sets. So there's no fatigue, there's no human fatigue, and the thing as well is that machines are able to pick up things that we call subvisual features or subvisual signals. So things that are like image texture, that are very abstract in nature. You could even go further, higher degrees of mathematical features that could describe that information that's contained within your image. And so sometimes those are the features that are actually more important than the ones that you just see with your own two eyes, like let's say nuclear size, for example.
Okay. Were there any like additional findings that you guys weren't looking for, but you maybe made a discovery about, or anything that really stood out while you guys were conducting this research?
Yeah, I would say the one thing that surprised me, or maybe wasn't too much of a surprise, but something that we did find was when we did a performance comparison against the gold standard of NGS, was we're looking at our data set, including those atypical patients and versus without those atypical patients. And the discrepancy between the accuracy of our models between including those and not including those patients was only about 5%. So that just goes to show that our model was very, very good at picking up these atypical cell clusters and making a good diagnosis on those.
In one sentence, what you would conclude about this research using AI and bile duct cancer, what would you ultimately conclude?
So I would say that AI absolutely has a place in going back and kind of improving the performance of these pathologists, right? It to me it's a constant feedback loop of how a pathologist or a doctor can improve the AI and how the AI can improve the doctor, right? So with our AI, we were able to improve the sensitivity of diagnosis because we were able to kind of definitively give a diagnosis to patients that were not given a definitive diagnosis by the pathologist.
Okay, interesting. Do you have like a rough number as to how many people may have been diagnosed one way, but you guys were able to reverse that diagnosis?
I could say off the top of my head, there was one patient that stood out in this data set, in the validation set that we used to test the performance of our model. There was one patient who was diagnosed as atypical on cytology. So the pathologist made an indecisive, had an indecisive decision, and our machine went and diagnosed that as cholangiocarcinoma. So it was positive for malignancy. But it looked very, very similar to other benign cases, right? So that's why it had the atypical nature. That's why it was the pathologist made that mistake in the first place. So yeah, that was just one off the top of my head.
What do you see as the next steps with this AI model that you developed?
Well, a natural extension would be to increase the number of patients. And actually, that's something that we're doing now, is a collaboration with Emory University in Atlanta, and the hospital network down there. Huge hospital network where they have an abundance of cases that we could use in our study. And then the rest of the paper I cannot discuss with you because I don't want to get scooped. And I'm working on something on that actually right now.
Okay, so you do have a next step established for being
100%.
able to for this, and then potentially even in the future, if there's success from there, being able to scale something like this to create that accessibility for everybody. Is that right?
Right, and say the number one issue right now in AI in healthcare is kind of implementing your algorithms into a clinical setting, and there's a number of reasons why that's an issue. There's three reasons, because if you've heard of the black box dilemma of AI, is where you have deep learning networks that are not interpretable, they're not explainable, and most importantly, they're not transparent at this moment in time, right? So you'd have to be able to kind of understand the machine's thought process of what the machine is picking up. How is it making this decision of what is malignant and what is benign? How are these features that the network picked up, how are they explainable on biology? So in a clinical setting where people's lives are on the line and we don't know what the machine is doing. And lastly, when we talk about transparency, we're talking about a model that's reproducible and that's not biased, and it has really clean data that goes into it, that's unbiased, and that is void of any artifacts.
So would your model need to be able to address those three features for if it were to be successful in the phase two of your studies, to be able to move to a wide scale clinical approach? Is that right?
Yeah, so any model, I would say, to be implemented in a clinical setting must have these three things down: interpretable, explainable, and transparent. And
Okay.
to this date, very, very few models have done this, whether it's in the private sector, like say Google is working on things like this, Philips, or at the academic level, the universities are working on. It's very difficult to get a model to do that, right? Actually, a fun fact: there's about if you go back to the COVID pandemic, there was a huge rush or a demand of AI algorithms to be used into kind of identifying patients that had pneumothorax caused by COVID. So you had patients who had pneumonia that was caused by COVID, and you had patients who had COVID who didn't have pneumonia. And there were so many different algorithms that were put out there. I think about over 400 algorithms that people published within the last 2-3 years, and none of them have been used in a clinical setting. Now you're gonna ask why? It's because it's because there was so many issues with the data that people were using, right? So many of the data sources that people were using were without even them knowing themselves, came from different sources, and some of those data sets that were spliced together using duplicate images, so that causes overfitting, for example. So that's a massive problem. So you're going to have a biased data set that can perform very well on a very specific set of images, but then when it goes and sees patients from a different site or from a different country, it starts to fail, right? So in turn, that model is useless.
In terms of the next phase of your study, how long do you see that being conducted for?
It's hard to say. The biggest timeline challenge is getting the data from an institute. It could take months. It could take years. There's a lot of administrative things and legal things that go between the transfer of data between two institutes. There's a lot of regulations that have to be complied with. So, for example, HIPAA, you need to fill out data use agreements and MTAs, and all of these processes take time. And to make
Okay.
sure, again, that your data is clean, and to make sure that you really specifically ask your collaborators, because, for example, my collaborator is a cytopathologist at Emory. I want to make sure that what I ask from her is not a waste of her time, right? And I want to make sure that pulling these cases from Emory, we really have a clear idea of what we want to do with the scope of our project, and to make sure that we don't miss anything, so that later when we go back and try to get some other information, nothing is missing.
My question is phase two. How large of a study is that going to be?
Oh,
Like how many patients are going to be conducted within that study?
so it's hard to say right now. I actually just had an email exchange last week with my collaborator. Um, we're hoping for around 200 cases. That's optimistic. And so if we can get 200 cases, we could really, really do a good job with validating our model. Because you'll look around in literature and you won't find that many studies with a large sample size for this particular study. I started this project, this thyroid project, I would say in 2021, early
Okay.
2021. It's still ongoing because there's a lot that can be done with this project. There's actually two or three different papers that I'm going to be pushing out for this project, and each one has a different goal in mind. Yeah, so the main project that I'm working on now, or the main paper that I'm working on now, is looking at the risk stratification approaches to the different subvariants of papillary thyroid carcinoma. So, in essence, to create a model that combines our pathomic and our genomic information to provide more insight into stratifying patients into different risk categories. So we can have high risk, low risk patients who belong to this subvariant of the carcinoma, and high risk and low risk patients of the follicular variant, of the classical variant, of the tall cell variant, which is the most aggressive variant, and really getting granular with the profile of the patient that we could build, right? So instead of just applying a one size fits all kind of treatment for let's say classical variant of papillary thyroid carcinoma, which is the most frequently occurring one, we can get really, really specific with how we tailor treatments to patients.
Wow, that's awesome. How many people have you utilized within the study for right now?
So this study is using actually a public data set that comes from the Cancer Genome Atlas. There's 507 patients that are in this data set, and they come from 28 different institutes around the world. So it's looking at really one massive data set that contains multi-site information. And
Okay,
so
so
all
so
these
ul-
patients,
go ahead.
yeah,
My
so
apologies.
all of these patients, they're all confirmed to be papillary thyroid carcinoma. But the issue is the quality control aspect of these public data sets, right? There's either a lot of missing clinical information from these studies, or the scanning procedure for these images are not up to par, so we have to discard some of these images, and it really does affect um, the impact of the pipeline that you can make, right? So if imagine if you can make a very clean and strong pipeline with 1,000 patients, of course it's much more intriguing and much more convincing to a group of pathologists than having one that's only trained on, let's say, 200 patients.
Okay. So with all of this, for individuals, because there are a number of words maybe individuals may not be familiar with. So ultimately, what you're doing is you're using current data set of individuals that have presented themselves with those particular types of cancers, right? Using genetic information and pathological information, meaning just maybe imaging or anything of that nature, right? And trying to go and predict um, pretty much the recurrence of that coming in of that cancer. Is that right?
Right.
Okay.
And so originally what we did was to create a model that was just based off of exclusively the pathological features. So we were looking at nuclear shape of the cancer cells, nuclear texture of the cancer cells. We're looking at tumor infiltrating lymphocytes and how they're scattered across the tumor microenvironment. Really, all of these primitive cell structures that occur within the compartments of these tissues, and using those features to be able to predict, or kind of give a time to recurrence for a particular patient. But then we have identified from our pathologically based features a set of genetic pathways that were associated with the proliferation of these cancer cells as well. And so that was interesting to see that we identified certain genes that belong to well-known pathways of these mutations that lead to, for example, tall cell variants or classical variants or more aggressive behavior. And so we took those genes and we retrained our model with those genes, and we actually got an improvement in our model accuracy.
Wow. Is
But
there
again, I
is
can't go in detail into information. That's something that I'm doing right now, and I can't discuss more about the methodology of that.
no worries. Now, in terms of is your current process before kind of the AI technology that you developed, is there a current process right now to be able to predict the reoccurrence of the cancers?
Oh, there's tons of papers right now on actually particularly this data set too on thyroid carcinoma because there's a lot of data out there. It's a very it's definitely not as rare as the bile duct cancer, but it's also very indolent at the same time. Not many people die from this disease. Really, the issue here is, or the clinical issue is that there's a lot of variability in the recurrence. So it could be from 8 to 25% of people recur, and we want to really uh, give a profile to uh, to the risk of recurrence for those patients.
Okay. So there's research being done, but is there anything clinically being done to be able to identify
Yeah,
these?
so um, uh, a lot of these, like I said, a lot of these studies are actually deep learning studies, and again, we go back to the black box approach. Well, these studies they don't really provide the explainability of really what's going on in those cancers, right? So you're able to come up with a prediction of what may constitute as a patient that could be of higher risk or lower risk, but why is that the case? And on the clinical side, can you incorporate those clinical features into your model as well, so that it's more digestible for a clinician or for a clinical oncologist?
Are you able to share with us about any particular findings that have come from this right now?
Yeah. So one of the main findings that we had, published a couple of papers on this, conference papers on this, last year and this year is to be able to risk stratify these patients on based on their subvariant type. So we have classical, follicular, and tall cell variants, to be able to identify the morphological differences between these patients and how those relate to the risk of recurrence. So we identified that if we kind of break down our model into subspecific models, where one model may be validated on just classical patients, just follicular, just tall cell variants, we were able to identify a set of patients that have totally different uh, risk of recurrence from one another. So that was one finding, and another finding is more recent, is the inclusion of the genomic data with our pathomic data, and how that improves the robustness of our model and the explainability of our model, but also actually improved um, the accuracy of our model as well, and of
Okay.
how it was
Now,
able to differentiate between high and low risk of recurrence patients.
over what period of time are you trying to go and measure the recurrence for?
Good question. So it typically takes a long time for this disease, this papillary thyroid carcinoma to recur, because it's not an aggressive disease in relation to other cancers out there. So we typically look at a study period of 15 years for our survival analysis. And so these patients, they could be from 1980, they could be from 2000, from 2010. Um, but we do typically look at a follow-up period between 15 and maybe even 20 years, just to see if the patient has recurred.
Okay, so you're looking at data from back then and trying to go and compare it with data from about 15, 20 years after. Is that right?
Yeah, so when we look at our TCGA data set, there's patients of all different ages and different diagnosis states. So maybe one patient was diagnosed at age 80, maybe 20 years ago, versus one that was diagnosed 6 years ago and they were 24 years old, right? It's the good thing about our statistical methods that we have today is that um, as long as we have a relative standpoint of when we start collecting our time, and from the time to that event, it doesn't matter what age they are, or how long back that patient had their cancer.
Can you tell me a little bit about how you're currently measuring, like what metrics are being used in your study right now?
Yeah, so one of the metrics we use to quantitatively measure the performance of a prognostic study like this is the concordance index, and another one is the hazard ratio. And so the hazard ratio tells us the probability of one risk group over the other in terms of the recurrence. So we were actually able to determine within our training set that includes all of the different subvariants of papillary thyroid carcinoma. We had a hazard ratio of about 5.8, which indicates that the patients who are at higher risk were 5.8 times more likely to recur than the patients who are lower risk. And then we split those into their respective subvariants, and we were able to actually identify a larger hazard ratio amongst the classical variants and also the follicular variants. So that's where the advantage is with our model, is that when we're able to go in and increase the resolution of our model, right, to be very specific with what we're looking at. We're looking at a very specific sub-disease of this cancer, and go in and improve the risk stratification tools for that particular subvariant.
Wow, that's really interesting. So, how would you measure ultimately success within this study of your model?
The ultimate measure of success is to be able to, again, put this into clinical practice. And I think we're still a long ways away, and as is everybody who's working in this field.
But even beyond going into there, how would you particularly measure success to be able to take this to the next level in this particular study?
Yeah, so if you look at, we could compare the performances of our model to other models for other cancers as well. Typically, if you're able to get over an 80% accuracy on your model after validation, so for patients that or for images that the model hasn't seen before, that's particularly pretty good, right? And so it's a number that a pathologist will say, wow, that is something that's an improvement on what we have right now, or that's something that's an improvement on just a standalone pathologist performance.
Based on the information right now, is it more effective than any current method of being able to identify recurrence of such cancers?
Well, there is no other method right now that really goes in depth with the subvariants
Okay.
that we're looking at, right? So this is a novel project that I've been working on, and it's something that is of extreme use, I think, in clinical practice.
Okay. Is there anything else that has stood out to you that you're like, look, this is something that's really important in terms of the data we've collected, or any other findings, maybe of incidental, that really provided a lot of value for you and your team?
Yeah. So actually, for the subvariants of classical papillary thyroid carcinoma, those tumors can look quite different from one another, from patient to patient. It's called tumor heterogeneity, right? So some patients, um, they may look different than the other, and uh, because of that difference in the heterogeneity of the morphological structures within the tumor, you could have a varying risk of recurrence for those patients as well. So you could have aggressive and not less aggressive variants of classical variants of this disease. So that was one finding that we found out that really stuck out, is to be able to go in and not just apply a one size fits all treatment for classical variants of this disease. We can really look at um, the different risk levels of this particular subvariant.
Wow, that's awesome. Now, in being able to determine the recurrence, what kind of value does that provide for the patients in comparison to being diagnosed at the time of having the cancer?
Right. So if you're able to provide a kind of a date or a timeline to a patient of when you expect this, or how you expect this the cancer to progress, it kind of it gives the patient, at least in North America, that the way that they treat their oncologists, or they train their oncologists, is that you can provide the patient with a timeline of what they expect within the next few years, or 30 years, or 50 years, right? And so to be able to provide a very more accurate predict or timeline for a patient is very helpful to that patient's quality of life.
Wow. So not can your technology provide just the possibility of recurrence, but can it also provide the timeline of recurrence of how soon it can
That's
occur?
exactly what the model has done. So it will predict actually the time to recurrence for any given patient.
Wow, that's amazing. And so if they're able to get access to this information before the cancer even forms, right, or before it reoccurring, um, can they start treatment? I mean, I know you're not a doctor and you're not able to provide particular information, but is there a way to be able to proactively prevent a cancer from occurring if they're able to know from it beforehand?
Well, in the case of papillary thyroid carcinomas, usually you would just follow up as recommended because it's so indolent, and usually at the very beginning stages of it, you won't see any issues with your health or any particular change in your quality of life. The cancer is very indolent and it's not spreading. It doesn't metastasize that quickly, and so follow up is typically recommended with your oncologist. And so it's always early detection is the best course of action for a patient. And if they do require a more aggressive treatment, so for example, if they had a tall cell variant of this disease, then they could know that right away, or let's say not right away, but they can know it earlier than what we could than what is available with the tools today.
Man, that's awesome. Do you know when? I mean, I know you're currently conducting the research. I know it is very hard, but with something like this, do you know ideally? So let's say if you're able to go measure success, right? It's able kind of the outcome meets that 80% threshold that you brought up at least, right? And it's able to provide value. What do you see as the next steps for this study beyond this phase?
Well, say the next steps that I'm working on right now is to expand upon the subvariants that we use in our study. So not just
Okay.
looking at classical follicular and tall cell variants, but to go into diffuse sclerosing, hobnail types. These are more rarer types of the disease, but they do exist. And to really incorporate those types of subvariants into our models would be one of our future steps. And also to identify new pathways with the inclusion of more genes that could be determined. We're looking at the robustness of the model. So how much how can our model, or what is the scope of our model in terms of how much information it can actively predict, given all the information that we give it. So we want to really expand the base of our model to include different types of subvariants of this cancer so that it can go out and predict more and more different types of patients.
How long do you see it taking to be able to build a robust model that meets kind of your expectations?
I would say probably a couple of years, to make sure that we get data sets from different institutes, to make sure that our data is again all clean and unbiased, and has a healthy mix of different subvariants. So we don't want to just skew our data set with 80% of classical variants, even though that's the most frequently occurring, right? We want to have a sizable amount of the more rare cases as well, so that our model doesn't fail when we go and train those.
How do you see a model like this that you develop? Can that model be applied to all different types of cancers? Like, tell me a
Absolutely,
little
yeah.
bit about that, but how it can really be scaled, aside from a clinical standpoint, but in terms of just a diagnostic standpoint and application of different cancers.
Absolutely. It's just think about AI in general. It's not just applied in healthcare. It's applied in financial sector, in just not imaging and text-based models. It's everywhere around us, right? And essentially, the most important thing is to adopt a standardized pipeline um, for creating these AI pipelines, particularly in the AI and imaging fields.
If we're able to go and utilize AI for different diagnostics, could there be more accuracy in diagnosis, and could this really be changing the way we diagnose cancer and other diseases?
Yeah, so actually that's what the entire focus of our research group lab is. We're at the Center for Computational Imaging and Diagnostics. And now it's based in Emory University. It used to be in Case Western. We look at we span through a myriad of diseases, not just cancers as well. We look at kidney disease, we look at heart disease, and all different types of cancers, like a prostate, lung, breast, and me personally, I'm taking the lead on the bile duct and the thyroid. We also look at oral cavity and oral pharynx, and we all really help each other in developing our pipelines. But more or less, they are built the same way. Right? There's a standard way of going through the process of collecting our data, making sure that our data is going through quality control protocols, and then mining that data using certain techniques that have already been available for the past 40, 50 years in AI, to extract those features that are needed to make our predictions or make our diagnosis or to predict response to a treatment. Right?
That's
So
awesome.
if you think about, yeah, so if you think about another advantage that AI has in this particular field is that, well, a pathologist's job is to repeatedly identify these hallmarks or signatures or disease patterns within their particular specialty that they look at. Like, so for example, if you have a GI, a gastroenterologist looking at colon cancer studies or colon cancer images, inter-observer variability is a real thing where you have a lot of variability in how they grade those cancers. It's called they use a scale called the Gleason score, for example. And AI has actually come in and been able to identify or give a definitive Gleason score for some of these cases that the pathologists were previously disagreeing on, right? So again, it's that combination of AI and pathologists giving each other, using the advantages of both of them, and really helping each other out, using the strengths of both of a machine where it's able to do a well-defined task very efficiently on very large data sets without any fatigue. And then also you have the interpretability and the explainability of a human, and to kind of tweak that model and say, yeah, this model makes sense.
How soon can we start seeing AI being utilized in diagnostics of any particular disease?
I would say, um, if you wanted to be optimistic, we would be looking at the next 20 years. We're right now not in a position to be implementing our AI pipelines in clinical settings, just because, again, we wanted to make, like I've explained before, we need to make our AI explainable, interpretable, and transparent, right? So, and there's a lot of these black box models that are coming out, and we don't really have necessarily the means of making them totally explainable and interpretable. So when I'm talking about specifically our deep learning networks that are used left and right, right? So sometimes there are clinical problems that that someone just throws a deep learning pipeline at it and say, our model works, but why does it work? And it's kind of like, if I use the analogy of throwing an elephant on us on an ant to kill it, right? It's absolutely not necessary for most of the cases, especially if you want to use a smaller model or a more tailored model that is more explainable, and it's not necessarily as powerful. We don't need to be using that kind of computational load to be answering very particular clinical questions that we want to address. So it's always important to have the clinical hypothesis in mind of what you're trying to do, and what the answer you're trying to the question that you're trying to answer.
Okay. Now, so we've talked about diagnostics recently. I want to touch just slightly on treatments. Can you tell us a little bit about how AI, I mean, I know you're focused more on the diagnostic side, but at least maybe based on your research or based on different companies or developments, since you're pretty involved in that field, how AI right now is being used for developing treatments of cancer or other diseases?
Yeah, actually, this is something that came out last month. If you go back into, I'd say 2021, if in Science Magazine came out with the breakthrough of the year, which was an algorithm that was able to predict protein structures in the body. It's actually something that's called AlphaFold. If you're not familiar with it, it was able to predict the structure of all the proteins in the human body, which is, to me, is just mind blowing how we were able to do that only just a couple of years ago, right? And there was a group that came out from University of Toronto that that was able to extend this usage of this algorithm, right? So, and looking at the purpose of trying to predict the structure of a protein gives a lot of insight into the function of that protein and how those cell-to-cell interactions occur. That gives a lot of insight into how diseases work on a molecular level. So not just the cellular level that I work at, but really, really going into the genetic level. So a group that came out, I think it was last month, from University of Toronto, they developed, and this still boggles my mind when I think of it, an algorithm that is used for drug discovery for liver cancer. Right, so they use the AlphaFold, algorithm to kind of identify a gene that was responsible for the proliferation of cells of these hepatocellular carcinomas. So it's a type of liver cancer. Right, so they were able to identify one gene and then predict a protein that would bind to that gene to inhibit that gene from proliferating the liver cells. So I'm like thinking, wow, so you're going to create an artificial protein that effectively stops this gene from malfunctioning and from creating these conglomerates of liver cells that are cancerous. Right? So so that's just an example of how AI can be used in the drug discovery sector. You know, it's not particularly in my field of expertise, but there's just so much that AI could be used to revolutionize healthcare in the future.
Wow, that's some
Yeah,
amazing stuff. Just
it's,
AI, how it can be used
and
in so many
even
different.
then, like with the advent of these language models like ChatGPT, um, there was another study that came from UBC, and so that's in Vancouver in British Columbia. And they actually came out with a model with 80% accuracy. They were able to predict the survival rates in some cancer patients, and that was just using the dictation notes from a doctor's charts. And so that was using a language-based model like ChatGPT to go in and read the doctor's notes about the clinical information about that patient. So the sex, the age, maybe the smoking status, the alcohol status, all these different clinical factors that um, go into identifying or trying to predict the risk of recurrence for those patients.
So Shayan, we talked about AI being used for diagnostics, for treatment. I'm curious, how do you think AI will go and impact clinical management of patients in the future?
Yeah. So to answer this question, we need to make sure that we develop our algorithms in such a way that helps us tackle the big challenges of the healthcare that we face today. Right? There's no use of just using using the AI for the sake of trying to answer or to try to do what we already know. It's to do what we haven't done yet. Right? So there's only about 21,000 licensed pathologists according to the AMA in the U.S., which suggests that we need to really help the patient load as much as possible, right? That's a very, very small ratio of doctors to patients that we have. And and I know that pathologist is not the most glamorous or the most like face value type of doctor that anyone is accustomed to seeing, but they do all of the behind the scenes work, so to say, of all the laboratory work and all the diagnostic and prognostic work that goes into disease management and clinical management. That's all done by pathologists, right? And they desperately need the help of not just their own of their fellow coworkers, but also with the help of AI as well. And so the type of questions we should be asking, you know, to help them out is, how can we make clinically applicable pipelines that tackle the unmet needs that are in this clinical space for any particular disease? Right? Are there extremely difficult diseases that the human eye cannot detect, or any particular morphology that can only be seen by a computer or not by a human? Can we better risk stratify patients to avoid undertreatment or overtreatment of patients if it's totally unnecessary to treat a patient? Right, so that was one of the goals of my thyroid project, was to avoid overtreatment of patients if necessary. Right, we want to be able to uh, combine the advantages of a human, which is the interpretable and explainable aspect of this kind of system that we put into place into guiding treatments to better treatment pathways, but also looking at combining the advantages of a machine, which is to handle big data better than humans and to do things tirelessly, right, without any fatigue. And it doesn't even stop at diagnostics and prognostics. Again, I talked about the dictation of notes that are passed on between residents within the electronic health record system, or the quality control of patient charts could absolutely benefit from a computerized logistical system.
We talked about cancers of the thyroid and as well as the bile duct. I know you're focused more on the diagnostic side, but since you're involved in that field of study, I'm curious if there's any type of clinical trials that are out right now or soon to be out for treating cancers of those two.
Well, that's a good question that you asked because clinical trials are the gold standard of validating any scientific experiment because they contain data from multiple institutes. Um, so you're not just limited to a population that has been taking data in a certain way. Not the same scanner, not the same staining protocols. You have different institutes, and you also have strict guidelines for the participants of who are included in those clinical studies. And so, because of that, you're able to avoid potential confounding variables. You're able to be very strict on the type of profile of patient that you need to include in the study. Off the top of my head, I think there's one clinical trial that's going on for papillary thyroid microcarcinomas, and these are a particular special type of PTCs that are less than 1 centimeter in tumor size. And so this study was focused on using laser ablation to percutaneously shrink the size of these tumors. So it's essentially using very high temperature lasers to kind of just completely annihilate those tumors. And so the goal of that clinical trial was to evaluate the progress of those tumors after 3 and 6 months. And then, of course, as a consequence of that laser procedure, you're going to have thermal damage that's affects the around the ablation zone as well. So we need to see kind of what's the trade-off between how much the tumor has shrunk and how much residual damage has been done by the laser.
We talked about a lot of different topics. We talked about AI, cancer of the thyroid, cancer of the bile duct, um, the clinical trials that are available. Can you tell us? Would love to go in here. Are there any finishing thoughts that you have about anything of these topics that we talked about today?
Yeah, if there's one main takeaway from what I've talked about today is that don't be discouraged about how AI is going to revolutionize healthcare, right? Don't think about is AI going to replace doctors? It's not going to happen. If anything, there needs to be an augmentation of AI and how AI and doctors can work together to improve. Really, what the ultimate goal is to improve the quality of life of patients, right? It's the ultimate doctor's oath, is to be able to treat patients and always serve a community where is needed. And of course, with the advent of digital pathology, we're able to serve the most underserved and maybe remote areas in the world that don't have access to traditional healthcare services.
Shayan, it was truly a pleasure to go ahead and have you share all this information with us, the novel approaches you're taking, utilizing AI. This is some amazing things, and it's something that we should all be. I'm very excited to go and see the type of impact this technology can make and the type of contributions you're making within the field of diagnostics. It was truly a pleasure to have you. Thank you so much for sharing with
Thank
us
you.
this information.
Yeah, I'm really excited to see the legacy of what the scientists like myself and my peers are going to leave behind for future collaborators in this field, because it's a field that only started maybe 15, 20 years ago, and it only has massive room for improvement, right? So if I have any advice for people watching and want to get into this field well, not only just to make sure that you have a very rigorous knowledge in computer science, math, and the willingness to learn about pathology and anatomy, but Also, to make sure that you you really stick to a rigorous, standardized process of how you collect data, of how you process that data, and always have the clinical problem in mind, with at the back of your head. Never lose sight of your hypothesis, and just don't do things for the sake of for the glamour of AI. Right? Just make sure that your AI has a very, very niche use in what you're trying to accomplish.
Those are some great final words. Shayan, thank you so much.